All right, so continuing to talk about the cell cycle, which again is like the life cycle of the cell. The first phase of this cell cycle, which is pictured here, we're going to fill it in, is the G1 phase, or first gap phase. The second is the S phase, or the synthesis phase. The third is the G2, or second gap phase. Then you have mitosis, and then you have cytokinesis. So this is G1 of the cell cycle. This is the first step in the life cycle of a eukaryotic animal cell. Then we have the S phase to follow it, followed by G2. And then if you look at this yellow piece here, you can see it's divided into a lighter band and a darker band. These are going to be mitosis and cytokinesis respectively. You can see how small in terms of time they take to occur that these two processes are compared to the G1, S, and G2. So the majority of the cell's life is spent preparing for these two stages, the actual division of the nucleus and internal contents, and then the division of the cytoplasm and into two new daughter cells. And again, just to refresh, mitosis is the division of the nucleus and the internal contents. Cytokinesis is division of the cytoplasm, where that one mother cell splits into two new daughter cells. And ultimately, as we can see here at the end, two new cells are produced. This one cell produces a new cell, and you have two identical daughter cells. So before a cell can even begin to start thinking about mitosis and actually dividing, it must do two things. The first of which is it must duplicate all its chromosomes. Okay, so for eukaryotic animal cells, we have 46 chromosomes, 23 pairs. So we must go from 46 to 92 chromosomes. Then we must produce in our cells a supply of organelles for the new daughter cell. So we have to duplicate all our organelles. So we have to create a second nucleus. We have to not a nucleus. We have to create a second ER. We have to create a second Golgi. We have to create duplicates of all our ribosomes, our mitochondria, our lysosomes. All of these organelles have to be duplicated. And we're going to cycle back at some point in this unit and talk about why the nucleus doesn't need to be duplicated. All of these things, the copying of chromosomes going from 46 to 92, the copying of all the organelles, making copies of every one of them for the new cell, all of these things occur in the G1, S, and G2 phases. Okay, you can understand now why these are the majority of the cell cycle, right? This is the whole cell. The cell spends its whole life preparing for mitosis and cytokinesis. And you can see why it takes so long. And ultimately, the G1, S, and G2, because that's a mouthful, we're just going to collectively call G1, S, and G2, we're going to call it interphase. Okay, so when I say interphase, you think about G1, S, and G2. You think about this large proportion of the cell's life, of the cell cycle. So this green arrow around the outside, which encompasses G1, S, and G2, we'll call that interphase. All right, let's break these phases down just a little further. G1, here's what happens in G1. The cell doubles in size. It gets bigger. It's making room for all the extra stuff that's going to be inside of it. The enzymes, cytoplasmic organelles like mitochondria, ribosomes, ER, Golgi, and other molecules all double in number. And then in the S phase, DNA is replicated. So DNA replication gets its own phase of the cell cycle, right? Because it's so important. We have to get that DNA replication spot on. Those 46 chromosomes getting doubled into 92, those additional 46 chromosomes have to be identical to the original 46, ideally. It doesn't always happen that way, though. And then in G2 phase, the cell assembles special structures within itself needed for cell division. In G2 phase, this is where we start to see that kind of mysterious organelle known as a centriole starting to play an important role. We talked a little bit about it in Unit 3, and now we see here in the G2 phase of the cell cycle, it's starting to have a key role. So when interphase, again, interphase refers to the G1, the S, and the G2, is complete, that's when the cell is finally ready for mitosis and cytokinesis. It has doubled everything, and once it begins to split apart and then the cytoplasm splits apart, each cell will have one copy of everything and be a fully functioning, healthy cell ready to undergo its own cell cycle, make its own new cell. All right, so let's look at mitosis. We've gotten all the way through the cell cycle, but we haven't looked at mitosis, the actual division event of the cell. There are stages. The first of which isn't really of mitosis, it's of interphase. We know interphase is comprised of G1, S, and G2 and takes about 90% of the cell's life to do these things. The nucleus is well defined. This is what a cell looks like in interphase. This is an illustration. So here's the nucleus. Here's all the DNA inside. There's our nucleolus where ribosomes are made. And here are our centrioles. So the nucleus, well defined, I can see it. And it's bounded by the nuclear membrane. That's the light purple region around here. Outside the nucleus, we have two centrioles, and the centriole's function is to, we're going to finally figure it out, organize microtubules into a spindle. Wait, what? What the heck are microtubules? Don't worry, we're going to see an illustration of them. Basically, they're just proteins that reach out like little arms, and they're actually going to organize the chromosomes for our cells so the chromosomes get pulled apart appropriately. And we'll see illustrations of it. I know it's abstract now, but just know that the centrioles help to organize the chromosomes during the actual division event. And as cell division, as we get from interphase, G1, S, and G2, and into the phases of mitosis, we're going to see a real change in these centrioles. There's our nucleolus, and then ultimately we have our chromosomes. So the G1 phase is a period of intense biochemical activity, right? The cell doubles in size, enzymes are doubled, organelles are doubled, everything is doubling. And then the chromosomes have duplicated during the S phase, and they appear just kind of like a jumbled mass of fibers, like you see here. They haven't condensed yet. They don't have that classic chromosome X kind of structure just yet. And then in the G2 phase, the cell assembles special structures needed for division. That's where these centrioles are going to come in and play a crucial role. Just to recap the G1, S, and G2 phase, all collectively known as interphase here, right before we talk about the stages of mitosis. And remember, interphase is the G1, S, G2, and it's the period of time preceding mitosis. It's the cell getting ready to actually divide. Now, mitosis has 4 individual stages. So once interphase is over, these are the 4 stages of mitosis. We have prophase, metaphase, anaphase, telophase, and we're going to find out what all those mean in the next screencast, folks. Thank you.